Why don't you tell us, what's your name?
Jackson, of course.
And Jackson, what do you want to be when you grow up?
A scientist.
What kind of scientist?
A scientist that does everything.
What's this that you have next to you?
Oh, it's a walker. It helps me walk. But sometimes I don't like to use it. And I would rather not have this. Yeah, that's what I would rather not have.
How come?
Yeah, it's like, I don't really like it that much, but I don't know.
Gotta use it, right?
I have to.
Passing on this disease is really, it weighs on me. I feel awful. I mean, I wish I could have rewound that clock and said, hold on before we go forward. Maybe I should get tested. So the guilt's there. We plan everything based on what he can physically do.
I feel sad that as a parent, you know, I can't just have a typical family day and family experience. It's very hard sometimes to go through the day-to-day, constantly wondering what's going to happen next.
Among thousands of codons and among trillions of chances, is this what has to happen? It only takes one mistake of deoxyribonucleic acid, one single codon to duplicate incorrectly to mess you up permanently. My life has plenty of ups and downs, but there's a scary thought that forever lurks in my mind. Perhaps one missense mutation, one point mutation, one massive disability. I am Jackson Epstein, and I was born with central core disease, a form of muscular dystrophy. I'm lucky the disease isn't neurodegenerative and does not affect my brain or heart like many other muscular dystrophy diseases. It is congenital myopathy disease, muscle weakness disease, present at birth, caused by mutation. A mutation is the random change of genetic material which can cause positive, neutral, or negative outcomes. DNA contains 4 different nucleotide bases: adenine A, thymine T, cytosine C, and guanine G. A matches to T and G matches to C, forming weak hydrogen bonds between the antiparallel strands of DNA. For my mutation, there are currently no cures. Some visible effects or phenotypes of my disease include scoliosis, muscle weakness, difficulty to walk, and allergy to general anesthesia. Some of these phenotypes are visible simply by looking at me, whilst others are visible by allergic reaction. A physically abled person would not express these phenotypes. My specific mutation lies on the 19q13.2 or 19th chromosome and was dominantly inherited. This is the only mutation I personally have related to my disease. I am lucky that mine is also non-progressive, so it will fortunately not directly harm me more in my later years. Some other forms of central core disease are progressive. The major protein is the ryanodine 1 receptor, or RYR1. But what even is the RYR1 receptor protein? Well, it is a calcium channel protein which the Ca2+ provides nutrients to the skeletal muscle of the body. I have a blockage problem of calcium in the protein, causing my muscle weakness and other side effects. In mutation terminology, the RYR1 protein fails to properly regulate Ca2+, and a blockage of calcium causes a lack of nutrients to my skeletal muscle due to a point mutation on the second nucleotide of the codon. My specific mutation is P.ILE4898THR. On the 4898th codon of the RYR1 gene, the intended amino acid for isoleucine was swapped with the amino acid threonine. Calcium blockage prevents sufficient supply of nutrients to the skeletal muscle, resulting in weakness. When looking at my DNA, the coding strand was supposed to be ATA to later transcribe into isoleucine. Instead, the coding strand shows ACA, the single point mutation on that second nucleotide, the 14,694th nucleotide in the gene, caused my entire disease. Central core disease, among many other burdens, are my outcome. Gene expression of central core disease all starts with transcription. Transcription, being the process of converting DNA into messenger RNA, provides a blueprint for protein synthesis. It occurs within the nucleus of a eukaryotic cell, such as in my own body's composition. Prokaryotic transcription occurs in the free-floating DNA in the cytoplasm. Transcription itself consists of 4 steps, where messenger RNA, mRNA, is created to be used as a blueprint for the next step. There are 2 major enzymes in transcription, helicase and RNA polymerase, which both help for the production of mRNA. First, the helicase enzyme begins to unzip or break weak hydrogen bonds between nucleotides of the DNA double helix, separating it. Next, the RNA polymerase enzyme attaches to the template strand of the split DNA and begins to add complementary base pair nucleotides. For me, that means the mRNA would build off of my mutated codon on the template strand, TGT. Third, the new mRNA strand is formed from addition of complementary nucleotides and separates from the template DNA strand. Complementary of the TGT would be ACA, changing the amino acid from threonine from the intended isoleucine. Therefore, my mutated mRNA is now ACA. Finally, the helicase rezips or rebuilds weak hydrogen bonds, connecting the 2 DNA strands back into a double helix. Transcription allows for gene expression, or for me, the expression of my mutation. Translation, succeeding transcription, is the process of converting mRNA instruction into a polypeptide sequence, a protein. It occurs within the ribosome of the cell, whose role is to synthesize proteins. The incorrect threonine adds to my RYR1 protein during translation. Where could have my isoleucine gone? mRNA feeds into the ribosome the site of translation in the 5 to 3 direction. Codons, found on DNA or mRNA, are read and interpreted in the ribosome, which one codon correlates to one amino acid. 2 codons are exposed inside the ribosome at one point of time. My mutated mRNA, being the codon of ACA, feeds through 5 to 3, as the first nucleotide is the 5 direction and the third is the 3 direction. A new form of RNA, transfer RNA or tRNA, is used to build proteins. There's also a third RNA, ribosomal RNA or rRNA, which is built into the ribosome. Each tRNA molecule contains one of 20 amino acids, along with an anticodon on the bottom to match with the mRNA. Anticodons are the complementary base forms of codons, which form temporary hydrogen bonds. The complement of the mRNA's ACA is UGU for tRNA, and that is what my anticodon would be. As one tRNA attaches to an mRNA, a second tRNA attaches to the next codon. A peptide bond then forms between those 2 amino acids on the top of the tRNAs. This forms long amino acid strands, also known as proteins. If you still can't grasp the idea of translation with a mutation, think about this. Imagine looking through a warped glass ball at an object on the other side. I am on that other side, and I just don't appear normal. This is because of an error, or for me, the error of my DNA, which altered the mRNA, which was translated as mutated by the ribosome. It is important to mention that mutations can also be obtained through ultraviolet light or carcinogens. The mutation I had was specifically a point mutation and a missense mutation, which means one specific nucleotide was altered, being point, and resulted in the change of amino acids, being missense. Well, folks, that concludes my story. What I hope that everyone can take away from my mutation is this. I'm not a normal, physically disabled person, and many others in wheelchairs may not be confined to the seat of their chair. I am fortunately able to get out of my wheelchair and walk with crutches, which significantly increases my mobility. Remember, people in wheelchairs are never just confined to what they sit in.